Okay, hello everybody. Good morning and good afternoon, good evening, wherever you may be. Welcome to week 4 in the fall iteration of this long-term animal research seminar series. This week we hear from Rita Fortuna and Dr. Joseph Travis. We'll hear from Rita first, and before I introduce her, I'll make a couple of announcements. First, if you're participating via Zoom, you should see a Q&A tab on either the top or the bottom of your screen. And if you open that tab, you'll be able to type any questions that you have, as well as see and vote on other people's questions. At the end of the talk, we'll go through those questions, starting with the ones with the most votes. But if you have a clarifying question that you feel like needs to be addressed during the talk in order for you to understand something, you can just type clarification in capital letters at the start of the question. And Rita and Joe have said that they'll do their best to answer those questions in real time. And finally, recordings of the talks are available on YouTube shortly after they conclude. So if you need to leave early, or if you know others who cannot attend live, you can watch this talk, or they can watch this talk as many times as they'd like after it's complete. And so now I'm happy to introduce Rita Fortuna, who is a PhD student at the Faculty of Sciences at the University of Porto in Portugal, where she has previously completed a master's degree in biodiversity, genetics, and evolution. Rita works with the long-term Sociable Weaver project, a team that she joined in 2015 as a master's student. Her PhD focuses on understanding both pre- and postnatal care and investment by mothers, fathers, and helpers in this species, and the consequences of those strategies for both offspring and maternal fitness. The common aim of her work is to better understand the benefits of cooperation and the causes and consequences of sociality. And today she's going to talk about her analysis of long-term data, but she's also done some really interesting short-term data collection and experimentation regarding acoustics and physiology in the system. So the work that Rita has done, I think, really highlights the power of long-term longitudinal studies to capture important within and between individual variation and reproductive strategies that's simply not possible with a cross-sectional approach. So with that, I'm pleased to welcome Rita and turn things over to her now.
Well, Matthew, thank you for that very nice summary. And thank you for inviting me to give this talk. I'm really happy to be here today. So I will start sharing my screen and hopefully you all see my presentation now. Hi, everyone at home. So my name is Rita Fortuna. And yes, today I'm going to tell you about maternal allocation in cooperative breeders and the specific case of sociable weavers and what we've learned so far. So about our long-term project, we all have this common aim of studying the causes and consequences of sociality. And for this, we use sociable weavers as our study species. These birds live in Southern Africa only, and our field site is in South Africa, as you can see in the red point. Sociable weavers are a highly social system. First of all, they are cooperative breeders, and if you're not familiar with this term, it means that there are additional individuals besides the parents that will help to raise the offspring. And to these individuals we call helpers. And in the case of sociable weavers, they are facultative because pairs may or may not breed with helpers. And they're also a very cool system because they are colonial and they actually build the world's largest nest. If you look at the photo, to these huge nest structures we call colonies, and then inside each chamber is where each group breeds and roosts. And I could tell you a lot of things about sociable weavers, but the most important to understand this work is that they are opportunistic breeders. So breeding seasons can last up to 8 months or longer if the conditions allow. And they're also relatively long-lived birds. And what we know about the groups is that the majority of the helpers are previous offspring of one or both of the breeders. We collect 2 types of data, mainly. The first one, survival data through annual captures that started being done in 1993 by a local ornithologist, and they were done until last year. And then we collect breeding data through intense nest monitoring. And in the study I will show you today, the first season I included was actually from 2008. So as you can see, the study started way before I joined, and the current leaders of the project are Rita Covas and Claire de Trelin, so the people we have to acknowledge for being able to work with these amazing long-term data and to have the funding for it. They're also my PhD supervisors together with Mathieu Paquet. And in this slide, I would like to acknowledge my main collaborators and co-authors for this work, but also everyone that has been involved at some point in in this project and helped collecting data or in the lab. So thank you, everyone. And so I'm interested in understanding how maternal breeding environments can influence reproductive allocation. And we know from ecological variables such as weather and predators, because of their influence on reproductive success, we can also expect them to influence maternal allocation. And we can expect that mothers invest more when the quality of breeding environments is better. And this can be especially true for species that breed several times during several seasons. So they have always this trade-off between current reproduction, saving enough energy for survival and future reproduction. But besides these ecological variables, there can also be social components that influence reproductive success. And this may be especially true in highly social systems like the sociable weavers. And if we think about the specific example of cooperative breeders, and we know that helpers bring additional food to the nests, we could also expect a similar correlation between maternal allocation and helper presence in mothers' groups. And so this could allow females to produce more offspring and when they are breeding with helpers. And even though this correlation was found in some species, it's not always found. And this is just an example of two species of the same genus where we didn't find, in one of them, we didn't find this correlation. But this doesn't mean that helpers didn't show an effect on how mothers allocate. And actually in super fairy wrens, it was found a negative correlation between the size of the eggs and helpers' presence. And this strategy is called prenatal load lightening. This hypothesis states that mothers with helpers will lay smaller eggs, so we'll save energy this way, but then the additional food brought by the helpers will compensate for this egg size reduction, and at the end we'll have similar fledglings in nests with helpers and nests without, which is actually often the case that we found in cooperative breeders. And so this theory and this strategy could help explain why, and so the benefit would be for mothers. This was actually studied since then in many cooperative breeders across taxa, and what we see from these meta-analyses until 2017 was that in most cooperative breeders, this load lightening, prenatal load lightening, was actually seen to be an important strategy. And if you pay attention to those points, number 3, they actually come from a study in sociable weavers. So in 2013, we found a negative correlation between egg mass and group size that suggested that also in this species, prenatal load lightening could be an important strategy for females. What we didn't know and we couldn't say at the time was whether this was a plastic response of each female to variation in her group size, because this was a correlative study among females. And the other thing we didn't know was whether we would always find this trend, so if it's consistent across environmental conditions, because it's a one-season study, and or whether we would only find it in specific environmental conditions. And from both theory and previous studies suggest that we may expect that climatic variables or other ecological variables modulate helper effects. So for instance, in good conditions, you would see prenatal load lightening, but in bad conditions, these would not be advantages anymore. So helper effects wouldn't be seen anymore. And so with this work, we tried to answer these two questions. First, whether allocation differences in relation to number of helpers could represent plasticity, and then whether ecological factors modulate helper effects on maternal allocation. And for this, we sampled the number of helpers for each female at each of her breeding attempts. And we managed to resample over 150 females, which we then used to study within female adjustments in maternal allocation. And the two measures we used was the mass of each egg and the number of eggs each female laid. We studied natural variation in weather conditions, and this system is great to do that because it naturally varies a lot. As you can see, rainfall levels can really drastically change the landscape, but it can also drastically change the amount of resources that are available for these birds. But actually, the most important factor explaining reproductive success is snake predation. So these birds suffer really high rates of predation, and so we ran an experiment in which we wrapped trees with plastic, avoiding that snakes would climb up and predate the nests. And actually, our experiment duplicated fledgling success from each egg laid. So with this experiment, we were able not only to assess what's the effect of removing this pressure from snakes on maternal allocation strategies, but also to study if also this ecological variable could modulate helper effects in these species, as we predict for climate. So I will start by showing you egg mass results, and these models were ran with many covariates, so all the results I will show you are already corrected for all the covariables in the mixed effect models. And the first one I want to show you is that opposite to what we found in 2013, with all these data points, we now don't see any significant correlation between egg mass and number of helpers. And as I told you before, we wanted to look specifically at within females adjustments. And so for this, we ran a bivariate analysis using Bayesian methods that allowed us to partition the correlation between number of helpers and egg mass between females and within females. So it's in the within females mean that we will see if we can see any adjustments or not. And what we found was no evidence for a correlation between number of helpers and egg mass, neither between nor within females. So from all these results together, no evidence that females load lighten in sociable weavers. We also didn't find evidence that the number of helpers effect was being modulated by either rainfall or our predator exclusion experiment, meaning that the presence or absence of this helper effect is not determined by specific conditions, at least the ones we tested for. And actually, the single effects of these ecological variables didn't explain much variation in egg mass. The range of egg mass is quite wide, but egg mass was repeatable within females, and the strongest effects we found were of mother identity and mother size. In relation to clutch size, we saw that most clutches are of 3 eggs, and we modulated clutch size as a categorical variable. So what you will see in the plot is the variation in the probability of different clutch size categories in relation to number of helpers. And as you can see, there is no clear correlation between the two. So 3 egg clutches are more likely across all group sizes. And then when we assessed within female plasticity again, we saw a very similar result. So the mean correlation between number of helpers and clutch size was not credibly different from 0, both between and within females. So again, no evidence that females are adjusting the number of eggs laid in relation to number of helpers in their group. Some things went according to what we expected, and this was the case for the relationship between clutch size and rainfall in this case. And as you can see in the plot, the probability of having 4 egg clutches significantly increased as rainfall levels increased, and the probability of 3 egg clutches decreased. And this was also the case for our predator exclusion experiment. So in protected colonies on the right, again, the probability of 3 egg clutches decreased and the probability of 4 egg clutches increased. So summing up, we haven't found any interaction between our social variable and ecological variables. Number of helpers didn't seem to explain variation in egg mass or clutch size, and this was true both between and within females. On the other hand, ecological variables seem to explain variation in clutch size mostly, that correlated significantly with rainfall levels and the exclusion of predators through our experiment. So to answer my 2 questions, females do not seem to benefit from the presence of helpers, either by load lightening or by laying more eggs when they have more helpers in their group. And we also found no evidence that ecological variables could be modulating helper effects. What we did find was that egg mass was consistent within females and across environments, which is a common result in avian species. And on the other hand, clutch size correlated with our proxy for resource availability and expected breeding success through our predator exclusion experiment. So my take-home message would be that had we not done this long-term analysis, we would still consider that prenatal load lightening is an important strategy for this species. And even among cooperative breeders, maybe if we exclude sociable weavers from that meta-analysis plot, we wouldn't see any more a general tendency across species. So I think this is another good example of the importance of long-term data to study these naturally variable environments. And it doesn't only allow you to study different contexts, but also to resample individuals across different contexts, which is crucial if you're studying plastic responses. So thank you for listening. And if you're still curious about my work, ask a question now or contact me later. I'll be happy to answer.
Great, thank you very much. That's terrific. It looks like we have two questions from Susan Alberts. We'll start with her first one, which is about the design. She asks if the plastic sheaths that exclude the snakes also exclude other terrestrial predators like small and medium-sized mammals.
No, they don't. They don't exclude these predators, so it's just snake predation and that's it. But they are the biggest predator for offspring in sociable weavers.
And then she also is wondering about the benefits provided to and by breeders. So she asks, do you think the helpers benefit by staying to help, given that the breeders that they're helping don't seem to benefit? Or alternatively, do you think there's some other currency, perhaps offspring survival, in which the breeders are beneficial or they're benefiting?
So as you understood from the start, this is what we're trying to answer, and all these people in this project are trying to approach it in different ways. But I think that there could definitely be indirect benefits for the helpers, because as you saw, most of the helpers are kin of these parents that they are helping, and also direct benefits. So we know that these birds roost together and forage together in groups. Their social bonds are stronger within these cooperative groups. So there could be also a bunch of direct benefits for these helpers to help.
And then maybe one more question from Mark Halber, who asks, is there a sex ratio bias in the embryos? And I'll tack on to that. Have you looked to see whether sex bias is influenced by presence or absence of helpers? I
haven't looked at it for sure, and I'm not sure that we have looked at it recently, but it's definitely something we want to explore. So sex bias in the colonies, and also because we know that females are the dispersing sex and males are often, most of the helpers are males. So of course, sex ratio bias could be an important thing to look at.
Well, thank you very much, Rita, for that talk and for the interesting results and terrific pictures. And if other people have questions, feel free to contact Rita directly. And now I'll turn it over to Liz, who will introduce Joe.
All right, great, thanks, Matthew. And thanks, Rita. I'm very pleased today to introduce our second speaker, Dr. Joseph Travis, who's a Lawton Distinguished Professor of Biological Science at Florida State University. Joe's research seeks to understand the feedbacks between ecological and evolutionary processes, and specifically he studies how populations adapt to local circumstances and how variation among individuals translates into birth and death rates that drive population dynamics. Above all, Joe's a great naturalist, and his work integrates scientific theory and natural history. He instills in his students, including me, the goal of identifying an interesting system in which to ask the right question. And because of this, his lab has worked on a diversity of systems, including fish, frogs, salamanders, birds, fruit flies, and even plants. Joe also works to apply his scientific expertise to societal problems, and he does this by serving on many advisory boards. Recent ones include advising the Florida Wildlife Commission and reviewing models for the demography of Florida black bears. Today, Joe will share with us some of his long-term research, which he works mainly on fish. So welcome, Joe, and I'll turn things over to you now.
Thank you, Liz. And thank you all for inviting me. I'm really pleased to be able to participate in this endeavor that you're having. I want to talk about the contribution of long-term research in organismal biology. And what I'd like to do is explore this in three parts. First, I want to distinguish a couple of different types of long-term research. Then I'm going to talk about ecology and adaptation in the Trinidadian guppy, which is the long-term project I will discuss. And then I want to talk about the challenges in long-term research. And I hope by structuring my talk in this way, I can provoke either with me or among yourselves some real deeper discussion about the value of long-term research and the compromises that it requires. So let me distinguish first the types of long-term research. I would distinguish two types. The first is the long-term study of many facets of a single system, population, community, and ecosystem, which is what a lot of senior investigators have done over a long time. So for example, I've spent over about 35 years studying varying aspects of the biology of this fish, Heterandria formosa, a small live bear in the southeastern United States. Everything from the additive genetic influences on population dynamics, this was published in 2000, more recently parsing out what density stress really means. How much of it is per capita food level, how much of it are social interactions. So in some ways, these two slides bracket the last 20 years of this project. The other type of research is this one, and I want to talk more and focus more on this, long duration observation studies or experiments. And that's really what I want to focus on here. Why would you undertake a long duration study? Well, there are two reasons. The first are what I call projects of necessity. Some questions require long duration studies if they are to be answered. The second are projects of opportunity. Long duration study will produce unanticipated results and generate new questions that were not even contemplated at the start of the work. And I realize that sounds a little optimistic, almost Pollyanna-like, but I assure you it's actually true. It actually is true. Now, no one's going to fund you to do long duration studies by just saying, fund me to spend 20 years looking at something because I'm sure something cool will turn up. And really, it will. That's not going to work. You have to begin with projects of necessity, and invariably, projects of opportunity will arise. I want to talk about one long duration study and the long-term research context in which it occurs. So I want to talk about ecology and adaptation in the Trinidadian guppy. I'm going to do this in three parts. I'm going to give you an introduction to the system. I'm going to talk about two projects of necessity and then three projects of opportunity. I'm not going to spend a lot of time on each project. In other words, I'm not going to walk you through all the data and all the evidence. I want to give you a flavor for each project. In the projects of necessity, why you have to do a long duration study to answer them. And in the projects of opportunity, how the combination of design and serendipity has allowed interesting things to happen. And indeed, interesting things that we never had anticipated, believe me. So let me give you an introduction to the system. And you may know something about it from my friend and colleague, Dave Resnick, and from some of the other papers he and I have written together. This is the Trinidadian guppy. This is a male at the back and a female at the front. They are live bearers in the family Pusilidae, and they, of course, live on the island of Trinidad, hence the name Trinidadian guppy. But we studied them in the Northern Range Mountains, which are here across the top northern tip of Trinidad. And there are three sets of drainages. There is the Caroni drainage, which is the lower left, the Orocooch drainage, which is the lower right, and the northern drainages. And they differ in that the northern drainages have a different fauna than the south-facing drainages do, but the system is pretty much the same, and I'll tell you what that is. The streams in the mountains of the northern range toward the top are the classic pool and riffle first and second order streams. At the bottom, they become rivers, some often very shallow but fast running rivers. And there are three main steady states, if you want to call it that, in these rivers. At the very top, you have nothing but killifish, Rivulus hardii, Hart's killifish. This is an omnivore, although by and large carnivorous, but an omnivore. The second community is one in which guppies coexist with rivulus, and the third community at the bottom is where guppies and some rivulus coexist with large predators, wolffish, astyanax, etc. The one at the bottom is we call it a high predation community, and there's a low predation community and a rivulus only community. these are separated in a striking way by what we call barrier waterfalls. And this is what we mean by a barrier waterfall. This one is about 4 feet high on the Rita River. These are limestone formations. The limestone has crystallized out and has formed essentially a dam. And the larger predatory fish can't really get over the dam, whereas the smaller ones can. Not that they leap over the dam so much as in the rainy season when the area floods, there are lots of shallow areas on the sides of the dam in which the fish can move upstream. Rivulus in particular can actually go overland. Like many members of that genus, you can keep them alive in a paper towel if you wanted to. And that's been approved by the Animal Care and Use Committee at my institution, so that's okay. So this is what a barrier waterfall looks like. Now the guppies look very different between high predation and low predation, and this is the first thing we set out to explain. This is a good photograph, but a good depiction. On the high predation communities, the guppies mature, these are males, the guppies mature earlier, at a smaller size, they are less colorful. At the low predation communities, they mature later, they are bigger, they are more colorful, and as it will turn out, they actually have very different food habits. So this is an example from the Rita River. The communities are also quite different. This is a non-metric multidimensional scaling ordination of the invertebrate communities in the 3 types of places, high predation, low predation, and rivulus only. What I want you to see is that there's quite a difference on these 2 axes between high predation and low predation, less of a difference between rivulus only. Now, is this a matter, are these differences a matter of, for example, are the differences between high predation and low predation, how much of those are innate, if you will? How much of those are the result of the change in composition of the guppies, the different types of guppies, the different phenotypes of guppies? The difference between rivulus only and low predation, how much of that is due to the addition of guppies and how much of it is due to the particular guppy that's there, whether it's a low predation or a high predation guppy? So the project was designed to ask 2 intertwined questions. First, what really causes the evolution of that low predation guppy phenotype? Second, to what extent does the evolution of that phenotype alter the community? And to what extent does the evolution of the low predation phenotype send reverberations through the entire community that can help create these kinds of differences? So those were the 2 questions we set out to address, about, gee whiz, 15 years ago. Now we use a lot of different methods to address these. So this is a little bit like a typical long-term research question. We have artificial streams that we built. We have, you can do experiments in whole pools at the top. You can remove all the fish from a pool and reconstitute that pool in its own way. You could put a little barrier above and below it so the fish stay there. You can even do experiments within the pool. And if you look closely, you'll see that on the left-hand side of this picture on the bottom left, you'll see a wire structure within which there's lots of algae. You have to strain your eyes a little bit, but there's a similar wire structure on the right. The one on the left is electrified. The one on the right is not. This keeps guppies out of the interior of the structure. And you can see that excluding guppies causes the algae to bloom dramatically. So you can do in situ experiments in 2 ways. We can do mesocosm experiments. And then the really insane idea is this one. If you really want to know how something evolves, and in the order in which the various traits evolve, why not do experimental evolution in nature? What we did was transplanted high predation guppy stock upstream into reaches without guppies, but with rivulus in a controlled experimental design. We took stock from the Guanapo River, climbed up over the waterfalls. This is on the Taruri River. We didn't actually climb over these. We drove to the top of the mountain and walked back down and transplanted them into experimental populations. Here's a closer look with the map as to what we did. We took fish from very down below the Guanapo. Here's Trinidad and the Northern Range. Here's the Northern Range here. Here is our location. And what I want you to see is that we've made 4 experimental populations. The upper Lavaha, the lower Lavaha, these were transplanted in 2007, or 2008, I'm sorry. The Kaiwal and the Taylor were transplanted in 2009. Within this design, within each pair, one of the two streams, we thinned the canopy to enhance primary productivity by opening it up to more light. So the upper lava hull was thinned, the lower lava hull was not. In this particular pair, the tailor was thinned and the Kaiwa was not. So that's the experiment. And the idea was we were going to transplant these fish and follow them demographically and genetically. Demographically, we could mark every single individual fish with elastomer. This is our marking scheme. And here's an example of what the fish looked like when marked. We have marked over 50,000 fish since we began. We can follow these individuals until they no longer appear in the population. Our probability of recapture is about 90%. And you can recapture them almost, not quite exactly where you found them last month. They don't get out much. So we can actually recapture these fish, which means we could get monthly estimates of adult survival, recruitment, and somatic growth. We also take a scale from each fish, or a couple of scales, and with 43 microsatellites, build a pedigree. These are the kinds of data we get. This is just one example of it. This is female parents. This is in the upper lava, the early part of the study. Female parents are on the horizontal axis. The different male parents are in the different covers. And what you can see is that we can figure out for all these females how many offspring were recruited into the adult population and who the sires were. In other words, we know for about the first 5 years of the study now, we know every individual's mother and father. We know for any given individual how many offspring were recruited into the adult population. And we can trace both their genealogies and quantify reproductive success directly with this method. Our assignment probabilities are about 90% for one parent, 80 to 85% for both parents. So we can do very, very well. One of the reasons we do very, very well is that these fish are easily recaptured. Our recapture rates are so high. So those are the kinds of data we take. And the idea was to use that introduction to watch evolution in action, plus to do 2 projects of necessity. So I'm going to turn to our 2 projects of necessity. And then I'll talk about 3 projects of opportunity. First, the obvious question, what drives the evolution of these guppies? And there really were 2 hypotheses that have been advanced in the literature. One were differences in age-specific predation, because high predation is, well, high predation. And here, the life history of these guppies is that they mature quickly at small size and they have a tremendously high reproductive effort. Low predation is the opposite. They take later to mature, they mature larger, they have much lower reproductive effort, they live longer. And this would make perfect sense with life history theory if adults were heavily preyed upon down here and juveniles were heavily preyed upon up here, or fecundity was reduced. It turns out this is not the case. It turns out that there's one other thing that goes on here. With high predation, you get low density. With low predation, you get high density. And we used our introduction to test whether it was density that was selecting for these attributes in the life history. So with this particular transplant, we transplanted the fish and we monitored their population dynamics. These are the dynamics over the first 100 months. I love saying that, the first 100 months, because we do have another 100 months underway. But these are the dynamics. The open canopy populations are in the dots. The intact canopy populations are the solid lines. What you can see, especially you can see very clearly in the open canopy populations, are what are seasonal oscillations in dynamics. The scale here is a little different, a little difficult to see, but I want you to see that we started with really less than an individual per square meter. And eventually, by the time I show you the data I'm showing you, at about 48 months, we were up around 3 to 4 individuals per square meter. So the density had really increased. This is the result of a common garden study that we published last year in the American Naturalist. And this is the difference in the age of maturity and difference in the length of maturity in our paired introductions compared to the control, which is the guanaco, based on common garden results. So for several years, we collected juveniles, brought them back to Colorado State in Cameron Gallenboy's lab, and actually raised them in a common environment to look at the genotypic value, if you will, in a common environment. And what I want you to see is that in these particular upper and lower Navajo, and we also did the guanafo control, so we had 5 different sets of populations every year. You really start seeing a change in 2011 and 2012, 36 to 48 months afterward when the densities have gotten high. The same is true in Taylor and Kaiwal, and the same is true for this is the age of maturity, this is the length of maturity. So the common garden experiments reveal, after 5 years, that at the very high densities, we finally see the evolution of later age and larger size of maturity. It's bolstered by these graphs, in which we graph the difference in age of maturity and length of maturity of the fish from the controls against the grandparents' density. So the higher the grandparents' density, these are 2 generations in a common garden, the higher the grandparents' density, the greater the difference. So this suggests that it is indeed density-dependent selection. We could not have done this without at least 5 years in the 5 years of common garden data, 5 years of censuses, market recapture. Further, let me back up a little bit. We can show that this is driven just by what life history theory suggests, that the effect of density is not on adult survival, but is on some combination of a female fecundity and juvenile survival. Those are the conditions under which selection should favor delayed maturity and larger size maturity. So we could not have done this without those long-term data. The second question, I'll focus on one of those. The second question, as you know, revolves around the role of guppies and the change in phenotypes in determining the community. I want to focus on just one part of that, which is the coexistence between guppies and rivulus, the killifish. Now we know they coexist. We can do surveys across the island, and we've done surveys across the island. Other people have done surveys across the island. We know the 2 species coexist, yet, when we introduce the guppies, and this is what this graph shows, this is from a paper by Doug Frazier and Brad Lamphere a few years ago, introducing the guppies, these are from our experimental streams, drives the density of the killifish downward. The closed symbols are in the closed canopy, the open symbols in the open canopy, and these are introduction minus controls. The take-home message is that you introduce guppies and killifish drop like a stone. If you do laboratory competition experiments, guppies, which are the smaller species, can outcompete killifish in the size range where they overlap, which is when the killifish are young. So all the data we have, those data suggest that guppies should exclude rivulus. Now we also know there's a certain niche shift. We've done stable isotope work that shows that as rivulus get older and larger, they tend to eat more terrestrial food, particularly termite alates and ants. Guppies eat more aquatically based food. But our analyses, that niche difference, that character displacement, if you will, is not enough to provide coexistence. And we know this because one of the things we did in this, inspired by the system, was develop this model, Ron Bassett, Tim Coulson, and I, predicting coexistence in species with continuous ontogenetic nip shifts and competitive asymmetry. The upshot, when you have 2 species who change their niche as they grow, and there is asymmetries in competitive ability, there are a lot of routes to coexistence. And one would be that if the species that is not the dominant competitor, the worst competitor has to shift its niche a lot with size in order to coexist. That model suggests, when we parameterize it by what we know about these 2 species, that this is the parameter range in which coexistence can occur. This is based on the laboratory studies of competition and mesocosm studies and simulations of niche shift with size. And what has to happen is the killifish have to shift their niche quite a bit in order for the 2 to coexist. Everything we know thus far says that the system is here. They shouldn't coexist, yet they do. What we are thinking and why we need long-term data is that coexistence here is mediated not by fluctuation-independent mechanisms, but by what ecologists call fluctuation-dependent mechanisms, storage effects, and relative nonlinearity. And we think that's true because if you look at the seasons, pronounced dry and wet seasons, we find that guppies do better in the dry season than the wet season. Here is mean survival, guppies in the filled circles, killifish in the open circles. Guppies do better in the dry season, killifish do better in the wet season. If you look at the far right as to somatic growth, guppies grow better in the dry season than the wet season. Killifish grow dramatically better in the wet season than the dry season. There's a similar pattern in recruitment. Guppies do much better in the dry season. Killifish, we think they actually do better in the wet season, but the data don't show that. And partly that's because it's very hard to quantify killifish recruitment in either season, just the nature of the animals. But we're working on it. This suggests that the seasonality could play a big role, and that we may have a situation in which the storage effect applies, in which the guppies will outcompete the killifish in the dry season, but the killifish do better in the wet season, and because they have long-lived adults, can hang on through the wet season. Another possibility is that there's what ecologists call relative nonlinearity, where the two species have different nonlinear responses to the abundance of the limiting resource, that is food, in which the solid line might be killifish, because in the wet season, the resources are quite limiting because the heavy storms scour out the food. And the dashed line might be guppies, and the high abundance of resources would be the dry season. The only way you can actually test these theories is by long-term data, where you can actually quantify these curves and actually examine over the long run the responses of the two different species to different abundances of resources and to different seasons. It's a project of necessity. So it appears that understanding guppy-killifish coexistence is going to require the same kind of long-term data, probably even longer, than we found in asking the question about why the low predation guppies evolve. I want to talk about 3 projects of opportunity. And as I mentioned earlier, the projects of opportunity are ones that one doesn't anticipate, but that in the process of studying the project, conducting the projects of necessity, you make new observations, you discover new things. And I would be remiss if I didn't point out that many people would call this luck, dumb luck. This is where you get lucky. You're there doing something else and you see something. I would counter that luck is not quite right. I would quote the great Branch Rickey, the great baseball general manager, who said, luck is the residue of design. And so others have said the same thing, in different, more eloquent ways, perhaps. But the fact is that if you are working on projects of necessity and you have your eyes open and you pay attention, the opportunity will present itself. I want to talk about 3 projects of opportunity. I talked about earlier, I'll talk about the first one. I mentioned earlier how we were in one of our projects of necessity was watching guppies evolve to later age and larger size and maturity. Now, it's easy to study males because males don't grow after maturity. So we had those, I showed you the common garden results. Let me show you, and here I show you the picture again, just to remind you what we're looking at. I showed you the common garden results. Here's the actual phenotypic results over that same first 36 months. This is the mean size at maturity in males. Remember that by 36 to 48 months, they were much bigger than the control. In actuality, they were no different from when they started, phenotypically. The mean size here at a very high density is not that different from the mean size here. And in fact, there's a period where it decreased. Well, since we had this pedigree information, we could estimate quantitative genetic parameters. And we could run various models to look at whether the breeding values actually changed. And if you run those models using all the data at once, you get an interesting result. This is the distribution of using breeding values, the mean change in size and maturity after 3 years. If you actually, yes, there's a question here. Let me look at that. I have a PI and mentor question about guppies. How do you keep track of who is doing what aspect of research? That's a very good question, and I will come back to that in my third part of the talk. Who's doing what aspect, and what's my work and what's someone else's work. Thank you very much. I'll come back to that if you can just hold off. What I want to point out here is that we run two types of models. One that does not account for subgroup effects, the effects of season and the effects of density, higher density, and another that does. And what I want to point out to you is that when you account for the subgroup effects, in particular, the inhibitory effect of density on somatic growth rate and size and maturity, you actually see the big change in breeding value that you will not see if you don't account for them. So if you just run an animal model, you will get a result that suggests that breeding values do not change. If you run this mixed model with fixed effects for season and density, then you can show that the breeding values have really gone up. And that's, in essence, not accounting for the environment hides your ability to detect the cryptic evolution of breeding values. And this project was one of opportunity. We had all this common garden stuff, and when we were writing up the paper and looked at the actual field data, we said, wait a minute, why isn't this working? We should be seeing these fish getting bigger. Now, it turns out there's a reason for that. And this work was done by graduate student Tom Potter at Oxford. It's our second project of opportunity. This is the pool and riffle structure. And we literally sat around one night at our rental house in Trinidad at our headquarters, and someone said, what's the density of guppies and riffles? And David Resnick said, they're not in riffles. And someone said, how do you know? Have you ever looked? Yeah, I look. They're not in riffles. I don't see them. No. Have you ever really looked? So we decided maybe we should look in a serious way rather than just assume. Well, this was interesting. These are natural populations. We actually quantified densities in pools and densities in riffles. The pools are the solid lines, the riffles are the dashed lines. And again, this supports the intuition many of us have, is that the guppies are much less apparent in riffles, much less abundant in riffles. The same was true in our experimental populations. Here's the density in pools at the Kaiwa, the lower Wavaha, the Taylor, and the upper Wavaha. Here are the densities in pools over those first several years, and here are the densities in riffles. One of the things you notice is that as the total density increased, more fish were moving into the riffles. Is it better to be in a riffle than a pool? Well, again, we realized, geez, we have this pedigree data, and we know where we catch every fish every month. Let's see if we can test this. Well, we know their reproductive success. Let's see what we can do. So the first thing you might see at the low densities, these are negative binomial regressions for individuals we had a good long lifespan and we knew where they were. It looks like reproductive success is a positive function of riffle use. So despite the fact that they're not in riffles very much, not that many individuals are in riffles, looks like they should be. Except the density in riffles is lower than that in pools. So since we know where every individual is caught every month, we can develop a measure that I call the experienced density, which is the average density in individual experiences over its lifetime as it occupies either pools or riffles. So instead of thinking of density as the whole population or subpopulation, let's think of every individual's density experience. So the experience density is the summation over all the months where we caught the individual. The density in the habitat where this individual J was found in month I, divided by its lifespan. So for example, this particular individual was found in a pool in January 2009. This was the density, here's the density. This is a sample, you can take the average, and this individual in its lifetime experienced the density of 2 square meters. Now, when you look, you realize that experience density is a decreasing function of riffle use. The more time you spend in riffles, the lower your experience density is. If you now take experience density and look at that, you find that both when the population density was low and when the population density was high, the number of recruits is a positive function here of lifespan, but here it's a negative function of experience density. As the density goes up, reproductive success goes down. This makes a lot of sense. This is what we expect to happen. And once you adjust for density, there's no difference whether you experience that density in a riffle or you experience that density in a pool. We had no idea we were going to do this when we started this project. Finally, we have seen 2 episodes of massive floods that not only scoured the food from these streams, but scoured the guppies from these streams. Guppies are often very good at handling the wet season. And here's David Resnick in the far reaches of this stream in a raging torrent looking for guppies. And the guppies do this. They generally go into the shallow areas out of the main flow. And their survival in the wet season is remarkable given the velocity of the water and the way all the invertebrates are scoured out. But we had 2 situations in which there were high mortality rates. Now, one was in 2015. Now, by dumb luck, pure dumb luck, this massive storm came through during the middle of our marked recapture on the lower lava. That means that these animals were in the laboratory and not out in the stream. All the other 3 populations were in the stream. Mortality rates were very high, and worse the further downstream you got. Taylor was almost wiped out, Upper Wallowa, High Wall, and Taylor. And this gives you a chance to look at catastrophic selection. Is there an effect? Is there something that happens in these streams different from the typical selection that goes on? So I show you 2 things. Here is the body size distributions from geometric morphometrics of females in the lower Wallowa, the survivors from 1 month to the next on the bottom, the casualties on the top. And here it is from the upper Wallowa. And we had visions, by the way, when we did this, of, oh, this is going to be great. This is like catastrophic selection. This is Bumpus's sparrows. This is Peter Rosemary Grant's drought. Oh, this is going to be great. We're going to find cool stuff. Yeah, well. In the population that missed the flood entirely, there's really no difference between the body size of the survivors and the body size of the casualties. In the upper Wallowa, there's a slight statistically significant difference. It's very, very small. In other words, nothing really happened. I mean, there was a lot of mortality, but it was not selective. And I suppose this is interesting. Non-catastrophic, or catastrophic non-selection. Not all the projects of opportunity produce the result you hope for. I want to talk about the challenges in long-term research, and Mark raised one of them. Who should do long-duration studies? And how do you keep track? Now, these are the names of some of the people who were involved in all the work I showed you. The names in gold are the people who are still involved. Vassar, Coulson, Resnick, and myself. Vassar, Resnick, and Travis were there at the beginning. Coulson joined us later. The names in green are graduate students and postdocs who have been on the project. The names in blue are other faculty members who were part of the project at the start. The orange are our project managers in Trinidad, Mark Charon, Andy Van Alst, and then Ian Patterson and Dalhousie doing the genotyping. What I want you to notice is that all the people in gold or blue were tenured faculty members. Actually, all were tenured full professors when we began this work. All the short-term work was done by graduate students. That is to say, they got the glory for the mesocosm experiments, the pool manipulation experiments. All the stuff that was published in the first 7, 8 years really were graduate student projects. And the reason is graduate students and postdocs need jobs. They need to do work that has a decided endpoint where the question is two-sided and either answer is interesting. Graduate students can't risk their reputation on 10 years, on a 10-year project that might just collapse. When we actually, 2 weeks before we were going to introduce the fish, we sat in Jim Gillam's living room in Raleigh, North Carolina. And someone said, what are we going to do if like the first wet season comes and all these experimental populations are washed out and lost? Maybe there's a reason there weren't guppies in those reaches. You could have heard a pin drop in the room, and finally someone said, we'll never get another NSF grant again. Now, you know, I was a tenured full professor. I could take that chance. Others cannot. So who should do long duration studies? Well, if you're going to initiate them and really wait it out, you really had to have some job security to do that. It's perfectly great to join them. For example, people like Joshua Goldberg, the graduate student, joined the project. He's been able to use our long-term data set in part of his dissertation. Same for Tom Potter. So who should do long duration studies? You have to really think about who can take the risk of not getting any data for several years or interesting data. And then once the project is established, who can join the study is a little different. How can long-term research that should be supported, not by supported? Clearly, long-term research programs are supported by a series of National Science Foundation grants. Long-duration studies are difficult to support. The reason is that while we all love to have long-duration studies, and we often lament the lack of them, to keep it funded, you have to come up with a new question every 3 to 5 years. A new question into which you can shoehorn the data you've been gathering all along. In other words, you have to have a new justification for the same old thing. Now, the way we do that is to keep changing our focus on the question, moving from evolution to ecology, always doing additional mesocosm experiments, pool manipulations, other things to focus on the question du jour, as it were, while making sure that that question requires the long-term census data that we've been gathering. And then finally, when is it time to stop? When is it time to stop? When have you had enough? When have you learned all you're going to learn? Now, at some level, you could say it's never time to stop. There are always new questions. And that's true. But when is it time to stop a long-duration study? Now, some studies have gone on for decades, the White Woods, the Great Tits and the Blue Tits, and they're still finding new things to do. The Long-Term Ecological Research Program, the LTER program, has been taking very long, has a number of long-duration studies. But it's always fair to ask, when is it time to stop? When do you reach a point of diminishing returns? And when should the funds be used to support other investigators and other projects? Well, that's my story. I hope that I've helped you think about the types of long-term research. I've told you something about ecology and adaptation in the Trinidadian guppy, projects of opportunity that arose while we were doing projects of necessity, and challenges in long-term research. I'll end with something about long-duration studies. And that is, and I'll do this while I show you a picture of our compound in Trinidad where we live. If there are projects of necessity that require long-term studies, someone ought to be doing them. And I mentioned to you that all the people who started this project were tenured full professors. And I said it as kind of a jibe of needing an insurance policy. If it fails, it doesn't matter. But that's precisely why people like us should do long-term studies, because if we don't do long-duration studies, no one can afford to start long-duration studies. If you're a senior person, you should do them, because otherwise, all of our knowledge is going to be based on short-term studies. And as Rita's work showed us earlier, sometimes the longer term tells us that the short-term studies were misleading. So the long-term, long-duration studies are important. And I think it's incumbent on those of us with a certain amount of job security who have the personality for it to undertake these studies for the benefit of the field. Well, that's my story. I thank you for listening. I'll stop sharing my screen. And I really appreciate the opportunity to chat with you. And I hope it hasn't been too boring for you.
Great, thanks, Joe. I thought that was really thought-provoking. Well, maybe let's start with Mark's question from earlier, which is, how do you keep track of who's doing what aspect of the research, and critically experimental field manipulations with quote, your study populations across?
So we established a rule pretty much early on, which is nobody touches the experimental populations without, written consent of the network, as it were. I mean, nothing. The only thing that goes on in those populations is the census. And if you want to sample fish, everybody has to agree that it's worthwhile. And violators will be shot. Now, other than that, it requires extensive communication among a lot of people. So some of our friends, like Andres Lopez and Steve Thomas, are still doing ecosystem stuff, and they use the focal streams, though we are not doing that, we core group. And we just ask them to tell us when they want to come, and so we know, so we can regulate who walks through those streams. We also have very tight rules about what you can, we have dedicated nets for the streams. Nothing but the dedicated nets can be used so we don't bring in parasites. No foam boots. You know, I mean, we really have a set of very strict rules to protect those populations, but it requires a lot of communication among people because we're happy to share. We just need to, we just need to know if you guys, like when Andres and Steve want to work, can you please do it while we're doing the census so that we disrupt those populations only once a month? And we get along pretty well. With respect to who does the experiments, the mesocosms, again, that's really a matter of all of us working together to say, well, who wants to do it? Whose student is interested in this? And we really do work as a team. We're constantly Zooming nowadays. We're always constantly Zooming. And we really do work as a team to try to make sure that everybody's students get two-sided questions and get something out of it. I mean, does that help, Mark? Does that give you a good answer? Yes, no?
Well, I thought it was a great answer. We'll assume it's a good one. A comment rather than a question from Susan Alberts, I think, is worth. Mark says thank you. Yes, worth reading is she says thank you for a great talk and a thought-provoking talk. I found your discussion of different types of long-term studies illuminating and useful in thinking about my own research, and I completely agree with your summary point about the importance of people doing long-duration studies. And I want to push you just a little bit because you asked that question: when should they end? And I don't think you quite gave us an answer. And so
I did.
And so what what do you think the answer is?
Well, I think the answer is if you cannot come up, first of all, if you cannot come up with a project of necessity, in other words, if you've gotten pretty much the answers, and you're doing nothing more than dotting I's and crossing T's. If you're out of ideas, it's time to stop. It really is time to stop. And sometimes you need the reviewers of your proposals to tell you you're out of ideas, because there's a temptation to, having invested X number of years in a project, there's the we want 50,000 fish. I mean, if we just had another 50,000, what would be wonderful? And then you say, what are another 50,000? Who's going to tell you that the first 50,000 didn't tell you, for God's sakes? Well, you better have a good answer. The problem is you get so invested in it that sometimes it's easy to lose perspective. It's easy to sort of find yourself asking questions of your long duration study that only 3 other people are interested in. And 2 of them are like your spouses. And so they're not really interested, they're just humoring you. So I think you have to really say, if you don't have a new idea, then it's time to stop. I mean, yes, there will always be opportunities, but there's a point at which, if your projects of necessity are not strong enough, why don't we give other people the opportunity? Because funding is finite.
Yeah, I think that makes a lot of sense. I'll just note that Susan Alberts said, please don't say after 49 years, which happens to be the duration of the Ambacelli Baboon project.
Yeah, no, I mean, I think, when I look at the stuff in Whiteham Woods, I was there on sabbatical last year at Oxford, and I got to chat with Ben Sheldon and some of the people at work, and they continually come up with new ideas. I mean, they're not doing what David Lack did in the 1950s. They're not doing what people were doing in the 1970s. They're doing some of that, but there's really interesting quantitative genetics, paternity studies, behavioral studies, all kinds of neat things. And I think a really good long duration study generates those ideas. That's why I meant when I said if you keep your eyes open, opportunity arises. The opportunities are there, and you can turn those opportunities into projects of necessity.
Yeah, and I think I would just add that, having gotten to observe the Ambacelli project, it's like when endocrinology data and paternity data get introduced, or new techniques get introduced, a whole range of questions that not only did nobody think about asking when the project was started, but nobody asked 5 years before they could get the data, right? Like there's things that we can't even predict now we're going to be able to do in the future too.
I think that's exactly right. I mean, you have opportunities that you never conceived of when you started the project.
Well, this has been great. Thank you to you both Rita and Joe, for 2 really interesting and illuminating talks today. And if anyone out there has additional questions for either of them, feel free to get in touch. Otherwise, we will see you all in the same time and place next week. See you all later. Thanks again.
Thank you.
Thank you.